Ladies and gentlemen, thank you very much. For more than 40 years now, cold storage has been the standard method for organ preservation. And it helped by significantly retarding cellular metabolism. It prevented intracellular acidosis and it reduced edema. However, meanwhile, organ quality has dramatically changed. The donor age has increased, the adipositase in donors has much higher frequency, comorbidity is very often encountered, and an ischemic injury, for instance, upon DCD graft retrieval, is also frequently encountered. Thus, to sum up, organ quality and functional reserve of the livers are more and more critical, and this leads to a change of concept from simple storage to a more dynamic approach of organ preservation, including conditioning. This has led to a revival of the old days, as we have heard before. The machine perfusion techniques are refocused on. Here we see three commercially available examples: normothermic machine, hypothermic liver machine perfusion, and hypo or normothermically perfused liver in this machine. Why may machine perfused livers function better? And one of the first first parameters is the aeration of the organ. Also, cooling down the organ has slowed down metabolism. It is only slowed down and not completely interrupted. Therefore, always a little part of anaerobic metabolism is continued during cold preservation, and this may lead to an energy deficit and intracellular dyshomeostasis of signaling processes. Upon reperfusion then, depending on the gravity of these intracellular dyshomeostasis, we have a reperfusion injury, and that may eventually also culminate in cell death. Here we can have an illustration on the importance of oxygen during liver machine perfusion. It's an experimental model in rat livers, and machine perfusion overnight has been performed with no artificial oxygenation, with oxygenating the circulating perfusate with room air, that means 20% oxygen, or with pure oxygen of medical grade. And surprisingly, you see that only the 100% oxygenation led to a low lactic acid level in the perfusate at the end of the machine perfusion period, while already room air equilibration did not prevent lactic acid accumulation, and no oxygenation at all found significantly higher values of anaerobic metabolism. Translated into function after reperfusion, we see on the right side that bile production of the reperfused livers was significantly and much higher after 100% oxygenated machine perfusion as compared to 20 or no oxygenated machine perfusion. Why is this so? One, at least partial explanation, may be seen in a heterogeneity of microvascular perfusion, especially in hypothermia. Here we see a picture taken from a publication of the group from Lee. Initial hypothermic machine perfusion, as visualized by a fluorescence marker, showed homogeneous distribution of the perfusate in the liver sinusoids, while after extended times of hypothermic perfusion, we see that a lot of sinusoids are at least temporarily virtually unperfused. And as you may suspect, unperfused areas means non-oxygenated areas. If we have 100% oxygenation, we may speculate that from here, via diffusive processes, there is a diffusion oxygenation of these sinusoids as well, while this would not be able if we have already restricted oxygen availability in the still perfused sinusoids. So very late hypothermic machine perfusion has been introduced into clinical practice, and this was achieved by the group from Guerrera in Northern America. He has used in the first study more or less homemade liver perfusion apparatus, pumping flow controlled at low pressure the hepatic artery and the portal vein. And what he found was that early allograft dysfunction, the complex index, basically focusing on transaminase release, INR or bilirubin elevation during the first week, was nearly significantly reduced if he used the machine perfusion as compared to historical cold storage cases. As to graft survival and patient survival, there were no differences, but already he could note correlation of the AST released during this ex vivo circulation with the later observable early allograft dysfunction incidence after transplantation. Later, a more professional evaluation of the results showed that early allograft dysfunction was somewhat reduced. Significant benefits of machine perfusion could be evidenced for hospital stay and biliary complications. Overall, we have to say that the livers used in this study, or at least the recipients, were less endangered. They had low MELD scores, so maybe in a higher risk population, the positive results of a machine perfusion preservation might even come out more prominently. However, to perfuse the liver, you have to encounter a lot of logistic problems. You have to move a machine to the donor. You have to transport the machine, which is much more complicated than an icebox from the donor to the recipient. And we thought about what would be if we use the machine perfusion at a biphasic tool after preceding cold preservation as usual. What we see here is that most of the damage encountered after transplantation is not definitively manifest during cold storage, but only prepared. Mitochondria are intact after as long as 24 hours in HDK solution in rat livers, but 2 hours after reperfusion, most of these mitochondria are blown up and lose their function. So the idea developed that prior to reperfusion, you might recondition grafts by a short-term in-house machine perfusion in the recipient center. And this was modeled again in a rat liver model, an experimental one. And we see the negative control group, long-term cold storage, the positive control group, short-term cold storage, considered a viable liver, and the attempt to improve the long-term preserved livers by 90 minutes of oxygenated HMP. And strikingly, you find that equal metabolic activity could be seen in the reconditioned livers than in the healthy, short-preserved livers. Concerning transaminase release, it's even more striking that the reconditioned liver had even less injury than the healthy, short-preserved livers after 6 hours. Similar attempts were made in the Zurich group by Dudkowski, who also introduced this principle in clinical practice, and we see a comparison of end ischemic, he denominates it HOPE, hypothermic oxygenated perfusion of DCD livers. Compared to unperfused DCD livers, we see significantly less transaminase release, and he achieved that transaminase release in the treated DCD livers was as good as in the untreated heart-beating livers. As to graft survival, it is not significant, but it was an impressive, seemingly improvement obtained by the machine perfusion treatment, which was executed as we has proposed earlier, only 2 hours after preceding cold storage in the transplant clinic. From the mechanistic point of view, we could add that hypothermic machine perfusion apparently reduces biliary incidences, as shown by immunohistochemistry here in a rat liver model. It may also have an impact on cellular activation, usually encountered after longer periods of cold storage. What we see here is the gene expression of ICAM1 after cold storage or after 18 hours machine perfusion or 2 hours machine perfusion at the end of cold storage. On the left side, on the right side, an immunohistochemical image showing stain of ICAM1 around the vessels, which was not present after machine perfusion. Similarly, to a lesser extent, the gene expression of MHC class II genes were not significantly, but apparently slightly reduced. In immunohistochemical staining, we found MHC antigen reactive cells in the cold stored group, but not in the machine perfused group. Another way is cellular signal. Further to the energetic recovery obtainable by aerobiosis, we have an enforcement of signal homeostasis in the cell. I show you, for an example, the cyclic AMP protein kinase A pathway, which is activated by aerobiosis, and we can see that oxygenated machine perfusion with 100%, same model as shown in one of the earliest slides, obtains a nearly normal activity of the protein kinase A, while 20% or no oxygen during preservation resulted in a much lower activity of protein kinase A. This kinase A is a key kinase which, for instance, activates phosphorylation of PET, which means you have an anti-apoptotic signal, which also influences the activity and phosphorylation of endothelial NOS, which is responsible for a positive vascular homeostasis, and has also an influence on AMPK phosphorylation, a key proteinase which results in cell survival pathway activation via autophagy. Here we see, for instance, the AMPK activation expressed as percent baseline. Again, only the 100% machine perfusion resulted in normal values, while 20 or no oxygen resulted in significantly reduced activity levels of AMPK and thereby activity of cell survival pathways. So now to a completely different thing, that means normothermic preservation. That is the alternative to hypothermic machine perfusion. The philosophy is making the liver feel not being out of the organ at all, which is a conclusive idea realized by the organ oximeter machine. It is normothermic, automatic, and transportable, although transportable is a relative term. As a perfusate, there is used erythrocyte concentrate diluted with colloid-containing infusion. A lot of additional substances like bicarbonate or prostacycline are admixed during the perfusion, and sensors can sense blood gases and flow and pressure. In experimental studies, the prototype of this machine has yielded very encouraging results. If you supplant the 20 hours of cold storage in porcine liver transplantation by 20 hours of warm machine perfusion, represented by the red marks, you see significantly less increase in transaminases after transplantation in DBD donors. In non-heart beating donation, the effect is a little bit more dramatic. You see here the very high levels obtained after simple cold storage. These are all non-surviving animals, therefore you don't see any blue bars in the later observation period. Then this method has been translated into a pilot study in the clinic by a group of around Peter Friend, published by Ravi Kumar. It was a phase 1 trial with the primary endpoint 30-day graft survival, and they failed to obtain a benefit in this primary endpoint. However, they were able to find a significant reduction of peak transaminases in the first post-operative week. A larger trial has been set up under the sponsorship of CEU, a randomization of 270 adult livers either to the Organox machine, which here you see the transportability if you have a large van, or a classical cold ice box. And it is not yet published, but preliminary results presented at congresses point to a significant reduction of the here now as primary endpoint chosen peak AST to nearly half the values obtained in the cold storage group, and also lactate after reperfusion was found significantly reduced, and the early allograft dysfunction, which is a derivative. However, and this is important, this normothermic machine perfusion does not take benefit after preceding cold storage, as is shown here. I have to hurry up a little bit, and the explanation why this normothermic machine perfusion should be done either directly or not is seen here. If you perfuse cold preserved red livers at 4 degree, 20 degree or 37 degree, you find a progressive increase in mitochondrial injury, along with biochemical knowledge that abrupt temperature shifts lead to mitochondrial alterations and a reduced oxidative phosphorylation. We have thought to mitigate this effect by a controlled oxygenated rewarming after cold storage. And here you see either normal hypothermic machine perfusion or initially subnormothermic machine perfusion at constant 20 degree, or a gradual increase in temperature during the 90 minutes of post-preservation perfusion. And to sum up, only the latter group with controlled rewarming showed a significant improvement in bioproduction of porcine livers upon reperfusion, while the abrupt subnormothermic reconditioning showed no benefit against cold storage alone, and the hypothermic machine perfusion showed less benefit than the rewarming. This has been translated into clinical testing, and we found that controlled rewarming resulted in a significant reduction of transaminase release during the first post-operative days. It translated into a seemingly better graft survival compared to historical controls. A late update showed that 2-year survival in this rewarming group was still 100%. To be used to predict ulterior graft function as normothermic machine perfusion, of course, as well. We see here cold ischemia time, taken as a usual indicator of gulf viability, only scarcely related to INR in postoperative course, while the glucose release during subnormal thermic machine perfusion after rewarming showed a neat correlation with hepatic function after transplantation. So in conclusion, and I hope I'm in these 3 minutes, there is a trend towards a more dynamic preservation of livers. You have to choose which livers necessitate this more dynamic effort and which maybe not. Generally, an aerobic metabolism is to be achieved and cellular signal homeostasis has to be preserved. Maybe an adapted preservation temperature by hybrid solutions, leaving logistics of transport and retrieval unchanged, and then doing some treatment in the implant clinic by in-house reconditioning might be the solution. And the critical step, if you don't opt for the complete normothermic machine perfusion, to our opinion, is the transition from preservation to reperfusion, which may be alleviated by this controlled rewarming. Adjuvant pharmacotherapy has not yet been investigated in large scale, but may become more and more important as you warm up the machine perfusion temperature. Thank you very much for your attention.